Hello everyone, myself Manasi Korgaonkar. In this video, we are going to learn about an emerging technique in genomic science. So genomic science basically involves study of structure, function, evolution, mapping and editing of genomes. So these involves the methods like sequencing and analysis of genomes by using some bioinformatic tools to assemble and analyze the function and structure of entire genomes. So one of the technique used in genomic science is ChIP-Seq, which means chromatin immunoprecipitation sequencing. So what is chip sequencing? So in this word, CH stands for chromatin, which means mixture of DNA and protein. And the IP stands for immunoprecipitation, in which immuno means antibody specific for this protein is used. That's why it is immunoprecipitation. So this method is used to analyze protein interactions with DNA. It is an antibody-based technology used to selectively enrich specific DNA-binding proteins along with their DNA targets. It utilizes the antibodies that selectively recognize the and bind proteins including histones, histone modifications, transcription factors and cofactors to provide information about chromatin states and gene transcription. So now let us move towards the working of chip sequencing. So how does chip sequencing work? So chip sequencing has following steps as you can see in this picture. First is the cross-linking of proteins to DNA. Then there is cell lysis, chromatin fragmentation by digestion or sonication based shearing, immunoprecipitation using specific antibodies, DNA purification and DNA analysis. So now let us understand all these steps in detail. So first step is cross-linking of proteins to DNA. The first step is cross-linking using formaldehyde and large batches of DNA in order to obtain a useful amount. So in this step, formaldehyde is used because it can readily permeate cell walls and membranes, resulting in efficient cross-linking, that is formation of covalent bonds between protein and DNA. So in this picture, we can see the cross-linking of proteins, and glycine is added to quench the reaction. The cross-links are made between the protein and DNA, but also between RNA and other proteins. So the next step is cell lysis. The cell lysis stage extracts the cross-linked protein DNA complexes from cells or tissues and brings them into solution. So in this picture we can see these gaps which indicate the cell lysis. At this stage, cellular components are liberated by dissolving the cell membrane with detergent solutions. Because protein DNA interactions occur in nuclear compartment, hence removing cytosolic protein can help increase sensitivity. The third step is chromatin fragmentation. In order to analyze protein binding sequences, the extracted genomic DNA should be sheared into smaller workable pieces. As we can see in this picture, these are the chromatin fragments. DNA fragmentation is usually achieved either mechanically by sonication or enzymatically by digestion with micrococal nucleases. So the ideal chromatin fragments can range from 200 to less than 1000 base pairs. The next step is of immunoprecipitation. To isolate modified histone, transcription factor or cofactor of interest, chip validated specific antibodies are used. So in this picture we can see the specific antibodies are used for that proteins to immunoprecipitate and isolate the target from other nuclear components. So the next step is of DNA purification. Before the specific DNA products can be amplified and measured, the crosslinks between protein and DNA are reversed. So this is typically done through extensive heat incubations or through digestion of the protein component with proteinase K. So proteinase K is used in this step because it cleaves at the carboxy side of aliphatic, aromatic or hydrophobic residues, and it also has high specificity Hence, it is used for removing proteins and nucleases during purification. So, to purify the DNA from the remaining proteins, phenol chloroform extraction method is used. So, in this picture, we can see that the purified DNA product is obtained. So, the last step is DNA analysis. The purified DNA products are quantitated by quantitative PCR, which can be achieved using the methods like nanodrop spectrophotometer, qubit fluorometer, or other spectrophotometric methods. So now let us see some applications of chip sequencing. So it is used in genome-wide profiling and study DNA binding proteins. It also locates the protein binding and interaction sites in a genome. It captures DNA targets for transcription factors or histone modification across the entire genome. It also helps in studying the gene expression and mapping of genes. One of its important applications is the discovery of new regulatory elements. For example, analysis of DNA binding sites in the mouse brain has led to identification of new tissue-specific regulatory areas of genes. So there are also some drawbacks of chip sequencing. As there is involvement of many manual sample preparation steps, there are high chances of contamination in this process. The second most limitation is the selection of antibodies, as it requires specific antibodies for proteins. Not all antibodies immunoprecipitate, and if they can, may not perform well for chip sequencing. Antibody selection has a significant impact on the success of this assay. The main disadvantage for chip sequencing is also cost and its availability. Chip sequencing also tends to have high numbers of unmappable reads along with a guanine cytosine content bias. So I hope you got a clear idea about chip sequencing, that is chromatin immunoprecipitation sequencing. So these are the references used for this presentation. Thank you.